Okay, so we're back on MATLAB with the same data set that I had loaded the other day. Remember, we had done a lot of things. We ran independent component analysis. We looked at these figures, and if you recall, when we ran independent component analysis, we did it on our entire data set. So what I'm going to do is I'm going to sort of clear the memory of some of this stuff. And before I clear all of this out, I do want to show you on MATLAB. If we bring up the MATLAB window, you can see that it did, as I discussed, take quite a bit of time to do the ICA. It actually took a total of 282 steps. Each step, I would say, takes 3 to 4 to 5 seconds, depending on how fast your system is. So it did take a while. I want you to look over here. Notice that we have this thing here called All EEG. Just going to double click on that, and then you notice that it tells me that All EEG is a struct file within MATLAB. It has 41 fields. It has things like file name, file path. Here's the number of channels. Here's the number of trials as it currently is stored. The number of data points, the sampling rate. You have a max and min. If you want to access an individual channel of data, that's found in this column right here. Notice it has 5 different sets. So let's pull up that. Let's pull up the EEGLAB interface, and you notice here I have 5 data sets currently in my active memory. So what that means is that data set 1 would obviously correspond to field 1, and that was our raw data, and so on. So until I get rid of those data sets, it's putting them in this struct. And to actually see the data, I would need to go to this data column. And if I double click on that, it's actually going to bring up all my numbers. You don't have to do that. You can basically call up all EEG.data, and you can store it in a MATLAB, either in a .mat file, or you can just give it another name by loading it into the active MATLAB screen. So what I'm trying to say here is that you can pull data from EEGLAB into MATLAB to do additional calculations. And that will be important later on in the project. All right, well, let's go back to the EEGLAB script. And I'm going to come back to the data set that was filtered but not pruned with ICA. I have had a couple of you ask me about, say that you try the filter and you got an error message. I don't know if that's because of the version of MATLAB or because of the version of EEGLAB, but I will say I've had that happen to me before. I resolved the error by doing a low pass and high pass filter separately. So like instead of 0.5 to 50, I just did a 0.5 on the high pass end, and then I did a 50 on the low pass end, and I just ran it through 2 separate filters. That seemed to resolve that situation. Hopefully that'll work for you. I think it has to do with how MATLAB, the version of MATLAB you're using, the operating system, and the version of EEGLAB you're using. I do know they improved the filter tool with later editions of EEGLAB. I just don't know what else they changed. All right, so what I want to do now is I want to close out all of my other windows so that we kind of clear this up a little bit, and then I want to go actually to this filtered data set, and we'll replot it just so I know I'm plotting what I think I'm plotting. And remember, we have not run independent component analysis on the version that we're going to look at. Actually, you know what? I may have just messed this up. Let's see. All right, so, okay, well, so we'll just work with data set 3, the filtered data set. And let's go back now and replot that data because I want to show you some things on the data. So you remember I showed you kind of the photos that we displayed to the person. So I want to look at these markers and I want to explain to you what has happened. Okay, begin is basically the EEG system starting itself. So it's just kind of an arbitrary point that you can, and the reason to do that, and I'll explain that in a minute. If you see this right here, this +1 MA, that is Mad Avatar, the first picture. And you notice that presents a little bit after the system is told to begin. Now, why would we put that delay in there? We put that delay in there in case you want to look at the data before the stimulus was shown. So in other words, here the system sort of syncs everything, and then there's a slight delay, and then the person is shown the picture on the screen of the Mad Avatar. We record their EEG data at this point, and you can see for this person it was like 2.4 seconds. They came in and they answered what they saw. So was it happy, sad, or mad? At this point they gave a response, hopefully saying mad. Then there's a couple of seconds that go by, and the system is reinitialized, and then you see over here, and let me just flip over a little bit. Here you see begin again. So we begin again, and then you see 2HR. So that is image number 2. It is a happy, real person. So M stands for mad, H stands for happy, S stands for sad, R stands for real, A stands for avatar, and C stands for cartoon. So if we go back to the EEGLAB window, kind of keeping this guy in the background, and we go to tools. You can ask it to, and there's several different ways to do this. You can do it through the edit menu, you can do it through the tools menu. Every time I do it, it's like I'm starting over because I just forget. But there are more than one way to get at this. I told it to select data, and here it gives me just basically a time range. I can select known time ranges. I can select different epochs. We're going to talk about what an epoch is in a moment. So let's cancel that. Let's look under the Tools menu at Extract Epochs. Okay, what an epoch is it is a selected time range of EEG corresponding usually to some given type of time locking event. So right here it says start, comma, end, and in MATLAB, remember, you can use a space instead of a comma. It's telling us the name for the new data sets. It has defaulted and added epochs. I'm going to tell it for this particular data set, we only want to look at happy avatars. And then I'm going to show you how to do that. Now the epoch limits, what it's going to do is it's going to start 1 second before the stimulus appears. That's way too much. I don't need to do that. So let me just go from, let me go from 0.1 second to 2 seconds. So right before the happy avatar stimulus appears, this is going to take 1/10 of a second of data before that. In fact, I could even make that 0.01. All right, so literally I want to try and time this just in the instant before, 1/100th of a second before the person sees the happy avatar on the screen. For the human brain, visually 1/100th of a second is a very, very short time, and our eyes cannot discern the difference. Why do I not start it right on the stimulus? No reason. And we could actually start it after the stimulus if we took away the negative sign. So we're basically going to get, for all intents and purposes, 2 seconds worth of data from the minute the person sees the picture until they respond. I'm going to cut that down because it looks like most people, well, let's just leave it at 2. All right, now you see these 3 dots here. So on these 3 dots, you want to click that. Oh, and look here, it's giving me all the possible stimulus events that people saw. And right now I'm interested in only looking at happy avatars. So that would be my HA. And then you can use your, on the Mac, it is Command Control. Let me deselect that. Okay, so I can use so I'm looking for all the happy avatars. So because there are different pictures, there are actually 4 different happy avatars that people see. So that's what I want my time locking event to be. So I'm going to tell it okay. And notice here are all my time locking events. And so yes, if you know what they're called, you can do this manually. So every time I have an avatar, it's going to pull the data from 1/100th of a second before this shows to 2 seconds after it shows. I don't know if it'll do 0. Let's see if it'll do 0. We'll see if it'll do 0. For some reason, I was thinking in the past that had been a problem. Right now, we're not setting out of bounds max and mins or anything like that. We're just going to let it default. So I'm going to tell it okay. And then it's going to come in, it's going to create a new data set. If you want to save it, and you should save it, then you save it. At this point, I'm not going to save it. You can save it here. And let's see, at this point you also have the option to edit your description. I'm good with everything, so I'm going to say that's okay. And now you notice it's going to ask me about a baseline latency range. That is referring to something referred to as an evoked potential, where you're assuming you want to 0 the baseline power in the EEG. For studies like this, it is typically irrelevant, so we're just going to leave it blank and say okay. And yeah. All right, so now let's come to our data set. You see it's updated, so I'm now going to be looking only at the data associated with the happy avatar stimulus, and let's plot that. So let's plot the channel data. Okay, and now you can see what has happened. I have here on my figure, here's the presentation of happy avatar number 1. It's going to record for 2 seconds. And by the way, the 0 did work, so that's good. So it starts right with the presentation. It records for 2 seconds. You can see the response here. So I'm actually recording data before and after they respond. Sometimes that's an issue because sometimes after people respond, they take a breath, sneeze, cough, move their head, blink their eyes, etc. And you can see some of the time there is associated with this response a little movement. Now remember, I took the data before I did ICA. So there are 2 possible approaches. One approach is to take your whole data set before you have done any selection of epochs or selecting only a certain time period of EEG around the presentation stimulus. So that's what we did in video 2, is we did the ICA on the whole data set. The other option is to do it once you have selected your epochs. So the advantage of that is that it's a shorter data set, and ICA is going to go much faster. And you have eliminated a lot of the bad stuff, or some of the bad stuff. Also notice that if we had really crazy stuff in our data, if you epoch your data first and you have one of the epochs that's just really bad, like in this case, you can see epoch 3 is really bad, I can actually remove that. And unlike if I'm working with the raw data, I'm removing the entire epoch. Now, why is that relevant? That's relevant because if you come into your data before it is epoch and you remove a really bad artifact, I'm just going to, while we're talking, I'm going to say this is epoch 3 removed. if you remove really bad artifacts before you epoch, then you've played around with the time stuff. So you may not have, for example, let's go back to my figure. Where is that? For whatever reason, it doesn't want to plot it, so I guess it did away with the plot once I rejected the artifact. So we'll just go back and we'll pull up my EEGLAB window and we'll take our demo set that has that epoch removed and we'll plot the channel data again. So, and this time you notice it has actually relabeled. So we have a new 3, the old 3 is gone. So it relabels. Once you remove an entire epoch, it relabels. Now, why would you want to do that? Well, if you have one particularly bad region of data, it definitely makes the ICA analysis much more difficult. All right, so we're going to start right here, and we'll do ICA again, because ICA is kind of hard. There's kind of a knack to it. Oh, and just for kicks, let's see how many total epochs we have. We have 15. Okay, so remember here that we basically are presenting the same kind of stimulus to this individual. We originally presented it 16 times. We decided one of the epochs was really noisy and we threw it out. So just out of curiosity, why am I not throwing like this and this out? If you look, you see how repeated this is. It has a similar shape. That's going to be an eye blink, and that's going to be something that probably ICA will take care of. All right, so we will run ICA here on this data set, and once again, we will default, and let's let it go. This one should only take a couple of minutes, so I'm going to try to keep recording. If it ends up looking like it's going to go a lot longer, and it does actually look like it's going to go a lot longer. So I'm going to stop the video here. Well, maybe not. It's kind of going pretty fast now. So you notice the shorter length and the fact that I got rid of some of the crazy noise has enabled us to step through much quicker, and we'll probably be done here in about 2 or 3 minutes. So just to summarize what we've done so far while ICA is running, we went to Tools, and we told it to extract epochs, and we told it we wanted to look for happy avatar. So one of the things that I'm interested in you guys doing this summer for your study, and I'm going to give you a few days to, or a couple days to think about what you would like to look at and give me some feedback. There are a couple of different kinds of comparisons we could do. For example, one thing you can do is you can look and see if for a given emotion, like happy, is there a difference in the response between an avatar and a human, or a real? So you could take all of the happy avatar epochs and you could take all of the happy real epochs and then you could compare them. Typically, statistically is how that is done. And you could see if there's any difference in avatar and real. You could also see if there's a difference per emotion. So you can do a comparison between a happy avatar and a sad avatar. And you can do a comparison between a happy reel and a sad reel. And you can actually do a two-way comparison using what's called analysis of variance. So there's a number of different kinds of comparisons that you can do. So to summarize what we showed the person, we showed them happy, sad, and mad. We showed them real, avatar, and cartoon. And then we asked them to choose. Okay, and then we asked them to basically tell us what the emotion was. We did this for a neurotypical group as well as an autistic group. So a third comparison that we could make is what is the difference between how an autistic person processes and how a neurotypical person processes. Okay, we're done. So let's look at component activations. So we've started over. We did ICA for the epochs. I want to point out one thing. If you're an electrical engineer and you have a little bit of background in signal processing, you understand that what we have really done is we have kind of like boxcar truncated the data from when the stimulus appears until 2 seconds later. So what that means is we've taken a data set and it's discontinuous every 2 seconds. That actually can show up as a background in your Fourier spectrum. If you imagine doing like a rectangular window and then concatenating data sets that are rectangularly windowed, at the boundary, you're going to get some odd stuff show up in the Fourier spectrum. Okay, so now let's also plot the head plots. So that's the component map in 2D. We'll do that. And we'll let it default. And hopefully what we get will be a lot easier to analyze and a lot easier to select our components. While we're letting these figures load, let's come back to this figure in the time domain, and we'll, it's trying to, it keeps trying, every time it reloads, it goes away. Let me make this smaller. Yeah, that way we can look at both. Man, it's kind of messed up my figure here. Yeah, I'm just going to have to be patient and let it finish. There we go. Okay, now we need colors. All right, so if you look at our figures, you can see we really clearly have this number 1. That's clearly an eye movement. You see this localized dot here. That's clearly some kind of noise. Probably this one is as well. This looks like it might be muscle movement. This looks like noise. This also looks like eye movement. So I'm thinking maybe the first, maybe 6 out of the first 8. Let me see if I can close this out and let's replot it. Somehow it lost itself a little bit. We'll replot the component activations. Okay, and then let's play around with our scale. Because we want to be able to see. Okay, so you remember all those eye blinks that were showing up? You see in this component 1, it looks like those are showing up there. And a little bit also in component 2. We obviously have something crazy going on here in, this looks like channel 5. And then probably also in channel 11. And you notice as I'm pointing to the line, this display down here is telling me what channel I'm looking at. So 1, 2, 5, 11. There's some weird stuff going on with 21, 22, and 23. So 1, 2, let's look at 3. Yeah, 3 looks kind of wonky as well. 4 is looking a little more average. But here's 8, and there's 8 on the time scale. And then what did I say? 11. Yeah, see, 11's got this weird thing. On the map, 12 does, but if you look down here at the channels, you actually don't see anything on 12. So I would say 1, 2, 3. Move this up so we can see 6, 11, and then these that are in the 20s. So let's go back to our EEGLAB. Let's go to Tools. Let's tell it to reject data using ICA. Actually, no, remove components. And so what did I say? 1, 2, 4, 6, 21, 22, 23, 11. Let's see, was there anything else? Move this guy over here so I can see my, maybe 39, 41. This one looks noisy. That's channel 29. So let's say 29, 39, and 41. So add 29, 39, 41. There's no right or wrong way to do this. A lot of it's just trial and error. Tell it OK. Tell it accept. It's going to remove those components, and then it's going to create a new data set where it has inverted that transformation matrix that we use for ICA, and it's now going to create a new data set. We will plot that. Okay, notice all those crazy big blinks and eye movements are gone. So that is a very good thing, because they have disappeared from our data set. We still have some noise. It's not perfect, but wow, is it ever better. Let me see if I can pull the previous plot. I think it's this one. No. Maybe it's this one. No, that's the components. Last one is hopeful. Yeah, here we go. Here was the before ICA, and here is the after ICA. And so you can see a couple of things. You can see that if we run ICA after we epoch the data, boy, do we get a better result, because it's just easier for it to do on a smaller data set. Okay, I'm going to shut the video off here now because I know it's long to listen to and long to process. And if I need to say anything else after I think about it, I'll create a fourth video.